In this video, I am discussing about the cytological basis of crossing over. Crossing over it is a very important process by which the variation occurs in the offspring generation. So, cytological means of diagnosis of condition through the examination of the cells. So, since the homologous chromosomes normally exchange reciprocal segments, resulting in the chromosomes will those exhibits the no morphological difference. So, therefore, except the visible chiasmata, there is no other cytological observation which will substantiate and provides the evidence that the actual exchange of the chromosome segments really takes place. So always the crossing over occurs in between the homologous chromosomes. So these two are the homologous chromosomes. Here and the chromosomes crossing over occur here. So after the completion of the crossing over here, there is a formation of the recombinants. So these two chromatids are recombinant chromatids, and these two chromatids are called non-recombinants. So usually morphological evidence of crossing over is presence of chiasma. So therefore, to demonstrate that crossing over is associated with the actual exchange of the chromosome segments, so that's why the spatial experiments were devised by the Kurt Stern in Drosophila and by H.S. Criston and B. MC Clinton in Carn. They conducted the experiment in order to prove the cytological basis of the crossing over process. So, in which those chromosomes were used in which their morphology was altered due to the chromosomal aberrations in order to make them identifiable from their homologo. Stern's experiments in Drosophila. Kurt Stern obtained a female Drosophila which possessed a heteromorphic pair of X chromosome. So one X chromosome is longer and L-shaped. Here you are observing. So this is a L-shaped chromosome. So and he crossed these two chromosomes. The second chromosome, that is second X chromosome, was shorter than the normal because a piece had been broken and translocated to other the chromosome. In this case, to chromosome fourth. So these two X chromosomes in a female fly could be distinguished not only from each other but also from normal X chromosome under the microscope. The shorter X chromosome carried two mutant allele, that is one recessive mutant for carnation eye, that is light eye color represented by the car, and the second dominant mutant for barred eye, so that is represented by the B. So the barred eye, so that is narrow eye, represented by the capital B. Here mutant allele means changed allele with the mutation process. The longer X chromosome carried two normal alleles of these genes, that is one dominant normal for red eye, so that is represented by the plus here, and the second recessive normal for round eye, also represented by the plus. So such females were crossed with the male flies having the recessive allele. For both of these genes, the CAR plus, that is a light eye color with a round shape. If no crossing over takes place between the two genes, the two types of the gametes, that is CAR B, that is light eye colored with narrow, and red eye colored with a round shaped eye, will be produced from the female flies if there is no crossing over process. If crossing over will give, actually crossing over occurs, so that will give the two additional types of the gametes, that is